you. So I will be switching now from the silviculture and the prescriptions in silviculture to the question how to evaluate habitat quality and function as we implement these prescriptions. And I will present to you specifically one monitoring approach to do that through passive acoustic monitoring. And for the purposes of the T3 watershed experiment, we're looking at using passive acoustic monitoring to evaluate the function of complex early-seral habitat, one of the prescriptions that Bernard described first in the previous presentation, and compare that to habitat created through standard silviculture approaches, standard regeneration as a result of intensive forest management. And let me start with description of the complex early-seral habitat. And this is earliest forest developmental stage, where forests regenerate after stand replacing disturbance such as fire, wind, and it lasts until canopy closes, which we say about 90% canopy closure in the stand. And it is characterized by remnants from existing remnants from the old stand, like standing snags and downwood, diversity of grasses and shrubs. And as I said, we'll try to emulate that through silviculture and compare it to the standard regeneration. So how are we going to assess the habitat quality of that prescription? So our objective is to compare bird occupancy and presence absence between complex early-seral and standard regeneration. And we will be doing this by using 10 songbird species as indicators. And we selected those to be common species because we need enough observations to distinguish between habitat quality that have similar vocalization activity, which is similar in terms of time of the year and time of the day, that have known habitat associations and small home range, and that's important for the particular monitoring approach, the passive acoustic monitoring. And in addition, for these bird species that we think will be good indicators for the habitat we're creating, we also look at them from conservation perspective. The USGS Breeding Bird Survey has reported that there is a decline in early-seral associated bird species by 2 to 4% per year. And then this population decline is linked to the reduction in complex early-seral habitat in the Pacific Northwest, and particularly in the coastal range. So what is passive acoustic monitoring? This is just one of the many monitoring approaches to detect animals, similar to traditional, alternative to traditional point count bird surveys, alternative to radio telemetry, wildlife traps, camera traps, eDNA, and so on. In this case, we're deploying an acoustic device and record the sounds from the surrounding environment. You see here one of our recorders. We strap it to a tree, and then you download the data back in the office. What are the advantages of this method? Is that it allows sampling very large areas. It's relatively inexpensive. It's non-intrusive. We don't call, we just passively record whatever makes sounds in the forest. It is a permanent data record, so that allows for us to revisit these audio files, and they can, to answer different questions, they can be repurposed for various objectives later. It's a rapidly developing technology, and there are advances both in the recorders, but also in the type of analysis that can be done. For example, an explosion currently of using these audio files for various sound indices, acoustic indices. And how we did the sampling. So I just want to step back for a minute and tell you that for the first portion of this study, We are now sampling all strata in our watershed, experimental watersheds, to compare the level of occupancy of the 10 indicator species among the 4 most prevalent strata. And after that, we will move after the harvest to compare the occupancy between the 2 ways of regenerating early-seral stands. So you can see the outline of the watersheds in black, and the 4 strata colored here. The canopy closure was almost non-existent, so that we currently are sampling stand initiation, 0 to 14 years, stem exclusion, mature forest, which is everything above 80 years, including old growth, and recently thinned stands, of which we have fair amount currently across the OESF. If we zoom in to see where the sound recorders are allocated, you can see these yellow blobs, and they are sampling in each of these forest strata, trying to avoid the edges of the forest, the non-colored area, as well as riparian buffers. So, in addition to recording sounds, we are also collecting habitat. You see here the layout of the habitat data, the layout of the habitat sample plot, which is centered on the recorder station. And we're collecting habitat elements that could be manipulated by silviculture treatments, such as tree species size, density, canopy closure, canopy lift and height, amount of dead wood, and several metrics for understory composition, the height cover of shrubs, as well as recording landscape characteristics later, mostly through remote sensing, known to be important to the indicator species, such as distance to edge, distance to water, and so on. So as of last year, when we started this project, we installed 2013 monitoring sites. We have 240 hours of audio recording at each site because we collected data for 10 days continuously. And so that amounted to more than 50,000 hours of sound recording for the intimidating 12.1 terabyte of data. And I'll show you, and I'll attempt to show you how this looks on the, what this data look and sound. Can you hear that? Okay, so this is a spectrogram of the species. You can see this is just a few seconds of recording. You can see the kilohertz here, and then the signature of the bird calls that we're trying to identify. So Teddy, how do we make...
Teddy, most people could not hear that.
Oh, well, that was just for illustrative purposes. Sorry about that. So how do we make sense of that data into, how do we make it into the input data for our analysis that really consists of what species were available, present or absent at certain, during certain repeated surveys, right? And that's a challenge. And just a moment. Okay. So the data processing, as with many technology-heavy monitoring methods, the field work is easy, but the data processing part is intense. And so in this case, we are processing surveys by viewing spectrograms that I showed you earlier and listening to audio. And there is a manual component where we validate and annotate target species, about 10% of the files. And also identify aircraft and rain events that can affect the detectability of the species. And that's something that Therese and Levi did this winter, and you heard their presentations. And then running an automated detection of indicator species by matching the calls. And you see here a calls of orange-crowned warbler, the calls and songs of the species. So matching those to our spectrogram to identify in which survey certain species was present. So our analytical approach will include occupancy modeling, and that accounts for both for occupancy, which is the probability of the species to be present at the site as a function of habitat characteristics, and the probability to detect the species if it is present, which is a function of certain measurable variables such as rain and time of the year. So we will enter the species encounter histories, the probability of detection, the habitat data that we're collecting both in the field and remotely, and generate a set of statistical models for each indicator species, and then select the best fitting models to identify which regeneration approach and associated habitat elements results in higher or lower habitat function, right? And you see here on one side standard regeneration, where we already have installed monitors. And that's an illustration from a different area of complex Early-seral, something that we're hoping to be able to emulate. Current status, as I said, we already collected data for first year, started on the habitats for the sound, started on the audio surveys this year. Habitat surveys are a little lagging behind, but we are collecting the data at various plots as well. We processed the data from last year and partially analyzed them. And after these 2 years, last and this year of pre-harvest passive acoustic monitoring, we are planning to have at least 3 years of post-harvest monitoring to compare the function of the regenerating sites. And with that, I would like to thank Lauren Keeney and Dan Donato, who are my co-conspirators on this project and very intrepid researchers, as well as Warren Devine for helping on various technical aspects of this project, Bader and Anchal for collecting data, habitat data, and Teresa and Levi for helping with the processing of data. This project is sponsored by Earthwatch Institute as there's a grant for citizen science project, and volunteers will start coming helping on this project this spring, and you can learn more